Hello students, let's continue with Mycobacterium leprae. Objectives: pathogenicity, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis and treatment. Pathogenicity. Now, Mycobacterium leprae is an obligate intracellular pathogen first identified in the nodules of Lepromatous leprosy patient. We have already talked on this in the previous lecture. Rod-shaped gram-positive organism that is acid fast when stained by Zell-Nelson. Okay. Lumps or bumps that do not go away after several weeks or months are seen. Nerve damage which can lead to complications with the ability to sense feeling in the arms and legs as well as muscle weakness. Fine. Symptoms usually take 3 to 5 years from being exposed to manifest within the body. However, some individuals do not begin to show symptoms until many years of the exposure. Okay. Long incubation period. Why? Because the ability to properly be able to diagnose when an individual came into contact with the disease is very difficult. Understood? Symptoms: discolored patches of skin, usually flat, that may be numb and look faded, lighter than the skin around. Growths, nodules on the skin. Thick, stiff or dry skin. Painless ulcers on the soles of feet. Painless swelling or lumps on the face or earlobes. Loss of eyebrows or eyelashes. Okay. Pathogenesis. Incubation period range between 9 months to 20 years. It replicates intracellularly inside histocytes and nerve cells and has two forms. One form is tuberculoid, which induces cell-mediated response that limits its growth, and the other form is I have not mentioned over here, I am sorry. Through this form, M. leprae multiplies at the site of entry, usually the skin, invading and colonizing Schwann cells. Okay. The microbe then induces T helper lymphocytes, epithelioid cells and giant cells infiltration. Okay. Next, the loss of sensation may develop as a result of invasion of the peripheral sensory nerves. Understood? Yeah, I am given. Sorry. The second form of leprosy is the lepromatous form, in which the microbes proliferate within the macrophages at the site of entry. Okay, first form was tuberculoid, second lepromatous. They also grow within the epithelial tissues of the face and earlobes. Suppressor T cells that are induced are numerous, but the epithelioid and giant cells are rare or absent. With cell-mediated immunity impaired, large numbers of M. leprae appear in the macrophages and the infected patient develop papules at the entry site, marked by a folding of the skin. Fine. Next, extensive penetration of this microbe may lead to severe body damage. For example, loss of bones, fingers, toes. It is very bad. Laboratory diagnosis. Primarily, it is diagnosed with the clinical symptoms. Okay, the predictive value is 98% in diagnosing leprosy. So the three signs which are used in diagnosis are hypopigmented or reddish skin patches with definite loss of sensation, thickened peripheral nerves, acid-fast bacilli on skin smears or biopsy material, means staining is done. Okay. Prophylaxis and treatment. Multi-drug therapy uses combination of antibiotics that kill M. leprae including dapsone, rifampin, then clofazimine, quinolones, azithromycin, and minocycline. Antibiotics must be taken regularly until treatment is complete. Or the M. leprae it has an ability to grow back. Preventive measure is to avoid close contact with infectious people who are untreated. Blindness, crippling of hands and feet, and paralysis are all effects of nerve damage done by untreated M. leprae. Treatment does not reverse the nerve damage done, which is why it is recommended to get treated as soon as possible. Okay. The Bacillus calmette-guerin (BCG) vaccine offers a variable amount of protection against leprosy in addition to its main target of tuberculosis. Understood why such vaccinations are important? Okay. Now, go through the videos properly, guys. Any doubts, any confusion, anything that I should repeat, just tell me. Okay. Start preparing. All the best. Thank you.